Welcome to the lecture on the regulation of gene expression. This is a 2-hour lecture that will feature prokaryotic gene expression during the first hour and then eukaryotic gene expression during the second hour. There's a lot of information here. It's a big set of information. The way to get through this is to stick very tight to the learning objectives and also to stick tight to the review guide. And that will help you make sense of the detail that will prepare you for the exam in this class, but hopefully prepare you for the MCAT as well. So here are the learning objectives for the first part. You'll see the focus primarily on prokaryotic systems. And of course, the second part here, primarily on the eukaryotic systems. So let's look at the regulation of gene expression in prokaryotes. And we'll be looking at concepts of promoters and operators and operons and RNA polymerases and activators and inducers. Let's see what we have. So a great place to start is to ask, why in the world would we ever want to regulate gene expression? Why? Well, we know that there are a lot of proteins that make up a cell. Bacteria, prokaryotic or eukaryotic, there's all kinds of proteins that have all kinds of functions. But we know is we don't need every single protein all the time. And so gene expression allows us to have expression of those genes to make those proteins that we need at a given time, and then not express the proteins that are not needed at any given time. So that's adaptability. And the best way to talk about that is if you're a bacterium and you're in a very fuel-rich and material-rich environment, you can bring amino acids in, you can bring fuel molecules in. There's absolutely no reason to synthesize those molecules. They are available. So you want to downregulate or turn off expression of those genes that are necessary to make like amino acids. But if a bacterium is in a fuel-poor environment and it must synthesize proteins, then it needs to synthesize its own amino acids, for example. So adaptability is going to be a big one. And of course, implicit in this is the conservation. It takes a lot of energy to synthesize proteins. So why use that energy if you don't need to? Make the proteins you need and not the ones you don't need. And then we'll see later for eukaryotes, a big deal for gene regulation is differentiation. There are ways to mix and match and edit and splice and change the RNA that comes off of the DNA, the pre-mRNA, and that can lead to a variety of proteins with similar structures and functions, but slightly different structure and slightly different function that allows the cell to have a very unique function. We will see there's a couple of strategies by which gene expression can be regulated. And it depends on whether you're a prokaryote or whether you're a eukaryote. And if you recall for the RNA synthesis, the transcription lecture we had, for a prokaryote, when the RNA synthase gets onto a piece of DNA and it makes the primary transcript, the mRNA, as soon as that mRNA starts coming off of the RNA synthase, the ribosomes will get on that mRNA and start making the protein product. So for prokaryotes, really, the only place that you can regulate gene expression is to regulate whether or not the RNA synthase makes the mRNA. So if a prokaryote needs a protein, it will make the mRNA. If the prokaryote does not need the protein, it will not make the mRNA. But we're going to see that if you're sort of in a condition where you kind of need the protein or not, there's going to be a thing called attenuated gene expression regulation, where you make a little bit but not a lot. So there is a little bit of variation there, but really it is the synthesis of the mRNA for prokaryotes. It is the primary site of gene expression regulation. Now, eukaryotes we know are higher organization. They've got a lot more sophistication. Here we know that the DNA is transcribed to RNA. The RNA is going to be inside the nucleus, and it will not be exposed to the ribosomes for translation until it moves out into the cytoplasm. And because the RNA will live inside the nucleus for a period of time, that allows the eukaryotic cell to do removal of introns or differential splicing or some kind of processing will take place to make an mRNA that can be sent out into the cytoplasm, which then gets translated and becomes a protein. So we'll see a variety of regulation of gene expression for eukaryotes that are simply not available to prokaryotes. Looking at prokaryotic gene expression regulation, a couple of fundamental terms come. First, of course, you know the gene. The gene is going to be that DNA genetic information that codes for a given protein. A gene codes for a protein. And from the RNA synthesis lecture, you recall that there is a promoter element. The promoter element is going to be a site where a factor will bind that will allow the RNA polymerase to bind, and that RNA polymerase will know where the start of the gene is and can make an mRNA transcript that faithfully carries the information of the gene. Okay, so we've had that before. Now, there's also an operator site. We've sort of seen it, but not really called it out before. An operator site will modify the activity of the promoter site. And by that, it means there may be a regulatory protein that binds to the operator that will directly influence whether the RNA synthase can bind or can't bind, or if it does bind, whether it will start transcribing or not transcribe. The operator is a control element, and that's what we're going to be focusing on primarily during the prokaryotic gene regulation part. So a factor is just a fancy word for a protein that binds to the operator or the promoter to change its activity. It's going to be a factor. For prokaryotes, it's the sigma factor. That makes it possible for the RNA polymerase to bind and transcribe. For eukaryotes, it's the TATA binding protein, very analogous there. Look at these factors a little more closely here. That's going to bring up the terms trans-acting factor and cis-acting elements. Okay, so trans means apart or different or separate. And in this case, there is going to be a factor, which is a protein that will bind to a bit of DNA, and it's going to affect the ability of the RNA polymerase to do its work. Now, this protein is going to be synthesized by a gene, and that gene may be somewhere else in the genome, somewhere away, far away, some other place, okay? And that's why it's called trans, because it comes from another place, away, different, separate here. So that gene will make an mRNA, that mRNA will make a protein, and that protein will then diffuse within the organism, within the cell, until it comes to its specific DNA binding recognition site. Okay? Now, what do we have? We have the RNA polymerase, which will bind to the promoter site, and we'll have the factor binding site that's usually called an operator site. Notice that these things are beside each other. They're next to each other. It's the same bit of DNA. And so that's going to be called cis. Cis meaning together or same side or together. So a transacting factor is produced by a distal piece of DNA, a separate bit of DNA, and it will bind to a DNA recognition site or a binding site that is part of the same DNA or beside, and that is a cis-acting element. Okay, putting it all together, let's go up one more step here. For prokaryotes, we made the point that prokaryotes will not often have just a single gene, but rather they like to couple together the genes for proteins that will work together to achieve some metabolic aim of the protein. For example, this may be the synthesis of an amino acid. You may need 3 different enzymes that will take this starting material and take it all the way to amino acid. So it doesn't make sense to make 1 or only 2 or only 3. You need to make all of those proteins so that you can make that amino acid. And so they will be grouped together as an operon, and that operon then will fall under one single promoter. The promoter now will be what allows the RNA synthase to bind, and the RNA synthase, once it starts, will move completely through gene 1, 2, and 3, and it will make one long mRNA transcript that will correspond to those 3 proteins. And of course, because these are gathered together in an operon, you want to have an operator element that will control the transcription of each of the genes inside the operon here. You don't care to make one or two or three. If you're not going to make one, you're not going to make any of them. So that's why the whole thing is together under that one operon. So you will have an operon that will make it easier for the RNA synthase to bind and transcribe all three genes, or you'll have an operator that will bind and make it harder for the RNA synthase to bind or to transcribe these genes. Now, what's also important is think about the prokaryote. When you have the prokaryote, as soon as the RNA synthase starts to make the messenger RNA, as soon as that messenger RNA starts to come off of the RNA synthase and it is exposed inside the cytoplasm of this bacteria, the ribosomes can get on that and immediately start translating. So you're going to see translation at the same time as you see transcription. And because all of the genes are present here in this messenger RNA, you're going to see gene 1, gene 2, and gene 3 all being produced at the same time. That's why it's important for prokaryotes that if you don't want these proteins, then you need to stop the production of that mRNA. And so the operator will shut down the entire transcription process. Here are the two major mechanisms for gene expression regulation in prokaryotes. That is negative control and positive control. And just like it sounds, negative control means a factor will bind to the DNA ahead of the gene that will prevent the gene from being transcribed. And for positive control, a factor will bind to the DNA that makes it possible for the gene to be transcribed. So negative prevents and positive makes it possible. Now there's two types of negative and positive control. There is induction and there is repression. So let's look at the negative side first and see how this works. Okay, so first what you'll notice for each of these four cases, there is going to be a protein that is produced, and that protein is what will bind to the DNA. And that protein now will be coded by an mRNA that will be produced by a gene that is different or far away. And because of that, this is now a trans-acting factor. That's what this is showing you in each of the four panels. There's a protein produced by DNA somewhere else. That trans-acting factor will have a recognition binding site that is in the operator, which is right before the gene of interest. So while it's not labeled, this is the part of the gene that normally would be transcribed into mRNA and then to a protein. So in the induction case here, what we see is the repressor protein is produced, and it will immediately come and find that operator. It will find that binding site, and it will bind with high affinity. And what you see here is no RNA polymerase. The repressor here will stop the RNA polymerase from binding and transcribing the gene. And that's fine. Now what you see over here on the right is genes under negative control are transcribed unless they're turned off by a repressor protein. In fact, that's what it is. This gene expression is turned off by this repressor protein. And so in order to get gene transcription, oftentimes you just have to find a way to release that negative control. And what the cell does, it will say, I want to release this negative control. And the cell will produce a certain molecule, depends on what it is, and it's right now whatever that molecule is. And when that molecule binds to this repressor, it will cause a conformational change. All those weak interactions and ionic interactions and hydrogen bonds and all that stuff will cause a conformation change. And when the protein changes its shape, it no longer has high affinity for the DNA. And so with the co-inducer bound, that repressor will come off. It's no longer active. So it's not shown in this picture, but in the absence of that repressor protein, the RNA polymerase can bind and the RNA polymerase can do the gene transcription. Okay? Now that's induction. You're inducing a change in the system that allows for the RNA polymerase to make the mRNA. For repression, it's just the opposite here. you'll see that the transactivating factor is made, but it's going to be made in a form that has a structure that has low affinity for the DNA. So it's not likely to bind in its native form. And the cell will say, I want to repress this gene. I want to stop this gene for a period of time. In order to do that, it will make another molecule. It's a different molecule, but it will bind to this repressor and cause a conformational change that now has very high affinity for that DNA operator element. So with the co-repressor bound, the repressor will bind and that will stop the RNA polymerase from binding and transcribing. So when this is an inactive repressor and has low affinity, it's not going to be bound to the operator, and so the RNA polymerase can bind and can transcribe the gene. So this is called repression because the gene is active until this negative element, this repressor protein, has that molecule on there and causes the thing to be shut down. Okay, so both of these are negative. Both of these are showing a transactive factor that are going to stop the gene from being transcribed. Either it's stopped initially and the co-inducer will remove that repression, or it will be produced but not be active until the co-repressor is bound, and that causes a conformational change that takes you back. Okay, we're going to see examples of these coming up pretty soon. The positive case over here is going to be just the opposite. Now, what this is saying, that genes under positive control are active only if the regulator protein is present. So for now, we'll say it's yes or no, on or off, this or that, black or white. We're just going to follow along. So in this case, we have a transacting factor that's going to be synthesized in a form that has low affinity for DNA. And so when it's first produced, it doesn't bind to the DNA. And according to this message over here, the RNA synthase and RNA polymerase can't bind and it can't transcribe the gene. And there will come a time when the cell says, hey, I need to make that product. So a co-inducer will be produced that will bind to this transacting factor. It'll turn it into a form that has high affinity for the DNA, and of course it binds. And as soon as that transacting factor binds, because it's a positive control element, it will make it easier for the RNA synthase to bind and start to transcribe the gene. Now, it makes it easier, it could allow it to bind. There's a couple different mechanisms here, but now the cell is making the decision by producing this molecule, yes, let's transcribe that gene. And of course, you can imagine the other opportunity here is that the transactant factor is going to be made in a form that has high affinity for the DNA. So as soon as this protein is produced, it's going to find its way to that operator element and it's going to bind right away. And that's going to allow the RNA polymerase to do gene transcription. And the cell may come and say, well, I want this gene to be on, but right now I don't want it to be on. And so it sends in a co-repressor molecule that will bind to that positive control transacting factor and cause a conformational change that makes it come off. And since our condition is, if that positive control factor is gone, then you don't get gene transcription. And so for a period of time, as long as this co-repressor molecule is present, that positive control element will not be present. So that's the 4 mechanisms, and I think that's really good for the MCAT exam and what you're going to need. Now let's add one little shade of reality here. This shows you on or off, yes or no, up or down, black or white, this or that. It shows you all or nothing. And what we know is that these things can have a degree. And so if the concentration of co-inducer is small, not very high, but small, you'll get a little bit of binding, but not complete binding. So you'll see a reduction in the negative control element, but not 100%. But if you have a very high level of this co-inducer, then you're going to have 100% binding to this negative control element. It will come off. And the same thing is true here. If you want to slow down gene transcription with this negative element, as the concentration of co-repressor rises, you're going to more likely bind to this repressor, and it's going to cause more binding to DNA. So there's a way that the bacterium or the prokaryote can say, I want a little more or a little less gene expression. You know, I'm going to block it a little bit or a lot, or I'm going to block it a little or a lot based on these 2 mechanisms. And the same thing is true here. You know, you could get a little bit more gene transcription or a little less based on the positive control side that you have. Let's show a couple of examples, and to do that we're going to use the lac operon, which was discovered and described for the E. coli bacterium, And you'll see this theme is pretty popular. And the lac operon is going to have two modes of regulation. The first part is going to be shown here, which is negative control induction, and then we'll do the other side on the next slide. Okay, so we know for organisms, glucose is the universal fuel. It's preferred. The bacteria will take glucose and do glycolysis all the time. It's a constitutive pathway. But the bacterium is not always going to be present in a high glucose environment. Sometimes it will be exposed to other sugars, for example, lactose. Lactose is the milk sugar. So not uncommon for a bacteria to run into some lactose. If you remember from the carbohydrate lecture, lactose is going to be a disaccharide. It's a glucose and a galactose. It's an alpha 1,4 linkage. Okay, so that's going to be fine. So what you notice here is that lactose is not bad because you get glucose, and the bacteria can use the glucose. You get galactose, which is a structural isomer, so it just isomerizes the galactose and turns it into glucose. That's fine, all right? But we have to be able to utilize that lactose, and it's going to require additional proteins to liberate the glucose and the galactose. And the lac operon now is going to contain the genes for these extra proteins that will allow us to use, or allow the bacterium to use, that the lactose. So under normal conditions, the bacterium does not want to use lactose. It doesn't want to, it wants to use glucose. So under normal conditions, you need to keep this gene silent or not transcribed. And so there will be a negative repressor here, an active repressor that will bind. And when it binds, it has a negative mechanism. It prevents the RNA polymerase from binding and going forward to transcribe these genes. All right, that's fine. But in the when lactose is present, a little bit of the lactose gets inside the bacterium. We'll talk about that in a minute. But a little bit of the lactose gets in there, and a lactose metabolite is formed called allolactose, and it's over here. Allolactose is an isomer of lactose. It's connected by a 1,6 linkage over here. And allolactose is going to be a strong inducer, and that inducer is going to bind to the repressor. And when it does, it changes the conformation of the repressor and makes it have a low affinity for the DNA. So up here, no inducer is bound, high affinity for DNA. Down here, the inducer is bound, low affinity for DNA. So as soon as that repressor comes off of the DNA, the RNA polymerase can bind and it can move through that part of the DNA, and then it can transcribe these genes. And it ends up making a polycistronic messenger RNA. If you remember, polycistronic means there is more than one gene. Each of these genes is going to code for a different protein, and each one is going to have its own AUG start codon. And the three enzymes that are here is beta-galactosidase, which is the enzyme that isomerizes lactose to allolactose so that you can have this signaling going on. Permease is a channel in the membrane that lets the lactose in, and then a transacetylase is going to attach an acetyl group to the sugar, which is part of the metabolism of the other sugars in the bacterium. Okay. So as long as lactose is present, and as long as allolactose is present, then you're going to have this co-inducer keeping that repressor off. But if the lactose level drops, or we'll see in the next slide, if the glucose level rises, then we're going to see a removal of that allolactose. We'll see this repressor go back to an active form, and it's going to bind again. The second half of this story is to consider what happens when glucose is available at the same time that lactose is available. And we're going to see the difference here about a glucose sensor that will control this process as well. Now, we're going to pick up where we left off in the last slide. In the last slide, we saw there was the repressor that would bind to this operator element and block the RNA polymerase from binding. But when lactose is present, you have this allolactose, which is going to be that inducer, which will bind and cause a conformational change, which makes that repressor come off of the operator element, and the RNA polymerase can lead to the transcription of these genes. That's where we left off from before. And in fact, that'll happen. You'll see the RNA polymerase doing its thing. Now, the other question is, if there is glucose available, and it's a fairly high concentration of glucose, the bacterium would much rather use glucose as its energy source. Glycolysis is the main energy producer for the bacterium. Glucose is the fuel. You don't want to use lactose when glucose is available. Why spend the energy to make these proteins? Why spend the energy to metabolize lactose when you have glucose? And the glucose sensor molecule is going to be cyclic AMP. So cyclic AMP is a molecule that accumulates in cells when glucose is at lower levels. When we do metabolism in biochemistry, you will see that cyclic AMP often goes opposite to what glucose is doing. We'll see how that works. Okay, so in the absence of glucose, cyclic AMP gets higher and higher and higher. Well, why is that important? Well, there's a protein here called the catabolite activator protein, or CAP. And this is actually a positive control protein that will bind to another operator element that will make it easier for the RNA polymerase to be activated so it will more readily and more rapidly transcribe these lactose genes that we see over here. And so if, in this case, we're looking at a positive control induction mechanism. When the transactivating factor, the factor here, it's a positive control, it's in a low affinity form. The cyclic AMP now is going to be the inducer that will bind, cause a conformational change, and that increases the affinity for DNA, but it also increases the affinity for the RNA polymerase. And this molecule will find its binding site, it will interact with the RNA polymerase, and that will activate the RNA polymerase to give you more gene expression because lactose is available, and that's going to be the primary source for the glucose that's coming in. So that works out pretty well. Now, what we said is the cyclic AMP is going to be responsive to the glucose levels. So when glucose levels are high, cyclic AMP levels are low. So this lower picture down here shows when glucose is high, you don't see any cyclic AMP. Because the concentration of cyclic AMP is low, it's no longer bound to this positive control element here, and it's gone back to its quiet or its inactive form. It no longer has high affinity for the DNA. It no longer has high affinity for the RNA polymerase. And so without it being bound, the likelihood of the RNA polymerase binding and being activated is much, much lower. Now that's in the presence of glucose, and even if lactose is available, it's getting into the cell, you're making allactose. Allactose is bound to this negative control element, it is off the DNA. But what this shows you is the positive control element is far more important for getting that RNA synthase located and transcribing than is the negative repressor from blocking it here. And so in the presence of glucose, in the presence of lactose, you're not going to get transcription of this gene because you have the glucose. The next example is going to be negative control with repression. And to do this, we're going to show the trip operon, which is genes responsible for the synthesis of the amino acid tryptophan. Now, just like bacteria need to bring in fuel molecules from the environment, they also need to bring in biomolecules that they need, and amino acids are important. And if they don't have those amino acids available in the medium, they're going to have to synthesize the amino acids. And tryptophan, of course, is one of them. It's one of the first well understood mechanisms here. And so the trip operon now is going to make a series of enzymes that are responsible for the de novo synthesis of tryptophan. So under normal conditions, you're going to say, yes, of course, I would very much like to make this amino acid. So you have the promoter site and you have the operator site. The RNA polymerase combined, it goes through. And as we saw before, it makes a polycistronic messenger RNA. There are individual information codons here for each of those enzymes. Each one has its own start codon. And if you make one, you need to make all. So it makes perfect sense. Now the question is, do you really want to make tryptophan all the time? Well, no, of course not. If you don't have tryptophan in the medium, if you're not bringing it into the cell, then yes, you really do want to make tryptophan. But if tryptophan is available, you want to block those genes. And in fact, tryptophan the amino acid is going to be a co-repressor that will work with this repressor molecule. And when tryptophan is present, it will bind, and when it binds, it causes a conformational change in that repressor that increases its affinity for DNA, and it allows it to bind to that operator element. And in fact, that's what happens. In the absence of tryptophan, the low affinity form doesn't bind, the gene is transcribed. In the presence of tryptophan, then you have the conformational change binding to DNA, and the RNA polymerase is blocked. And as we mentioned before, and we mentioned again, it kind of depends on how much tryptophan there is. If you have a lot of tryptophan, then you can imagine quite a bit of co-repressor bound to this negative control element, so you're going to be pretty well shut down for the gene. But if there's a little bit of tryptophan available, then you get a little bit of binding, a little bit of a block. And so again, the bacterium sort of can control its gene expression depending on what the environment is. But the next couple slides are going to show you a really nice fine-tuned way that the bacterium has learned to control the amount of gene expression. Okay, attenuation. Attenuation is a fancy word that simply, it's like a volume knob on your radio or your phone or whatever. Attenuation typically means to turn down. So if you attenuate something, you reduce the intensity. And that's what we're looking at here. We're looking at the transcription of these genes, and the attenuation is to give you more transcription or less transcription, kind of like a volume knob on your phone or radio or whatever. Now, to orient you before, this is the trip operon. This is that regulatory element that we saw before, and this is where that negative inducer bound, and when tryptophan bound to the inducer, it came off, but when there's no tryptophan, it went on. So that's where we were before. And of course, these are the enzymes that make tryptophan. Now this leader, this little bit of the DNA, is going to be a very special bit of DNA that's going to make an mRNA, and this mRNA is going to have structural regions in it that are going to affect whether or not a ribosome can bind, and whether or not the ribosome can bind is going to affect whether or not the genes continue getting transcribed. You'll see in just a moment here. Now it's important to say that the part of the DNA that is the leader has nothing to do with tryptophan synthesis, nothing at all. But it has everything to do with whether or not these genes are transcribed. So what we're going to notice is the messenger RNA that is produced from this bit of the DNA, it's going to have its own start codon, because this part of the messenger RNA is going to make a protein, and that protein is called the leader peptide. Each of these little circles is going to be an amino acid, and if you'll notice, there are 2 tryptophans in this part of that leader peptide, and the codons for those 2 tryptophans are located here in what we call region 1. Okay, so if you know that the ability to transcribe this DNA, to make these genes, to transcribe these genes, is going to depend on tryptophan. And you see here, there is a protein or a leader peptide that has tryptophan in it. That's the link. You will make this leader peptide when tryptophan is available. You will not make this leader peptide when tryptophan is not available. And that's the key. Okay, that's the key to attenuation. If you have a little bit of tryptophan, you make a little bit of leader peptide. If you have a lot of tryptophan, you make a lot of leader peptide. Okay, all right, let's see. Let's see how this goes. All right, here is the messenger RNA for that bit of the gene that comes from the leader sequence on the DNA. And there are regions 1, 2, 3, and 4. Right. Now, what we notice is that there are specific structural elements here that can cause the RNA to form a base pair arrangement. We know that messenger RNAs typically are going to be singular. They're not going to be base paired. They're going to be single strands. But it just so happens that the bases in section 2 are able to form base pairs with section 3. And if they're allowed to come together, you'll get what's called a hairpin loop. That just happens because of the nature of the bases. That's fine. All right. It is also possible that the bases for region 3 can form a double base pairing with region 4 here. Again, the nature of these bases is such that they will form that hairpin loop, okay? So the question is, which one forms? How does this work? Well, that's the secret. Here is this little box here, and this is going to be a ribosome, which is now on that messenger RNA. It sees that AUG codon, it's going to bind, and it's going to start translating the messenger RNA. And the question is, how fast does that ribosome move from the left side to the right side? And it has everything to do with these two tryptophan codons. When the ribosome is moving and it gets to these two tryptophan codons, if tryptophan is present and the tRNAs can bind, the ribosome will make the peptide and incorporate the tryptophan, and it will quickly keep moving down the mRNA. And as it does so, it's going to move into region 1 to region 2. And when the ribosome is on the part 2 of this, or region 2 of that messenger RNA, then 2 and 3 can't touch each other. Actually, the 2 and the 3 are going to be made at different times, and so the 2 is produced before the 3. You'll see it on the next slide. Okay, we'll see on this. Now, if tryptophan is not present, when the ribosome gets to those two codons, it's going to be stuck. It can't move forward. If there's no tryptophan, there's no tryptophan tRNA. If there's no tryptophan tRNA, it can't bind, and you can't get peptide synthesis. The ribosome is going to be stalled. And if that's the case now, the ribosome is only covering region 1. It doesn't cover region 2. So 2 and 3 can bind together. Okay, down here where the ribosome kept moving, you see the ribosome covered section 2. So 2 and 3 can't interact. Only 3 and 4 can interact. Okay, so here is a trick. Here's a trick. If 2 and 3 interact together, then you will get gene transcription. The RNA polymerase will continue to make the mRNA. But if the ribosome moves into region 2 and 3 and 4 together, that 3 and 4 interaction is going to cause the RNA polymerase to fall off. It's going to make it not. So when tryptophan is low, you do want gene transcription, and you will get gene transcription. When tryptophan is high, you don't want gene transcription, and so this arrangement here causes the RNA polymerase to come off. Okay, one more slide. It'll make sense to you, I promise. I hope. Let's see how this all works together. It'll be clearer here. Now, to orient you where we were before, there is the messenger RNA, the mRNA, and this is the mRNA that has to do with the leader peptide. So there is DNA, and the DNA has that leader sequence in it. That's why it's called the TRPL. The genes that make the proteins that synthesize tryptophan are much further down the DNA, and of course they're much further down this messenger RNA. So we're looking at the messenger RNA that makes that leader sequence we saw on the previous slide. And we saw region 1, 2, 3, and 4. And the point that was made in the previous slide is that 3 and 4 can bind together and form a very specific hairpin loop, or 2 and 3 can bind together and form a very different loop. And the RNA polymerase is going to be very sensitive to the presence of 3 and 4, but it's not going to be sensitive to the structure 2 and 3. So when 2 and 3 are formed, the RNA polymerase stays active. When 3 or 4 are formed, then the RNA polymerase gets kicked off. Okay, how does this work? What are we talking about here? Okay. So in this case, we have a high level of tryptophan. And when you have a high level of tryptophan, when the ribosome binds to the mRNA, it'll bind to the AUG, the start codon, and it will start to synthesize this leader peptide. Bump, 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 bump. As it moves to the right, it is synthesizing the amino acids. And when it comes to the tryptophan codons, because tryptophan is high, there will be tryptophan tRNAs, and those tryptophan tRNAs will go 1 and 2, and they will go in there, and those tryptophans will be incorporated into the protein, and the ribosome will keep moving to the right. So it moves and keeps making the leader peptide. Okay. Now, if you remember, for prokaryotic RNA synthesis, as the messenger RNA is synthesized, as the messenger RNA is coming off the RNA polymerase, as soon as enough of that messenger RNA is formed, the ribosome will bind to that messenger RNA and it will start to transcribe that will translate that messenger RNA into protein. And so as the mRNA is being synthesized, region 1 is synthesized first, region 2 is synthesized next, region 3 is synthesized, then region 4 is synthesized. Okay. When you have a high level of tryptophan, the ribosome is going to be moving fairly steadily to the right, and it's going to move into region 2 where that part of the mRNA is blocked. And because region 2 is no longer structurally available, as the mRNA is synthesized, you get region 3, you get region 4, and these two will bind together. 3 and 4 will find each other. They will form that the base paired hairpin structure. And as I mentioned before, the RNA polymerase will sense that structure, and that structure will cause the RNA polymerase to fall off. It will dissociate from the DNA, and it will no longer transcribe. And that ends there. So when you have high levels of tryptophan, you get no gene expression, and that's exactly what the bacterium is trying to get here. And it's only because the ribosome could move through the trip codons and it could keep going to cover that section too. When you have a low level of tryptophan, when the ribosome is bound to the messenger RNA and it moves through the region 1, it comes to those tryptophan codons, and because there's no tryptophan, there's no tRNA. No tRNA, you can't incorporate the tryptophan into the peptide. The ribosome will stall. It can't move forward without the tryptophan tRNA. But meanwhile, the RNA polymerase keeps moving to the right, and it keeps making the messenger RNA. And so first it makes 2, and then it makes 3, and then it makes 4. And since 2 and 3 come out sequentially in time, 2 and 3 will bind to each other. And because 2 and 3 are bound to each other, 4 cannot bind to 3. And since 4 and 3 can't bind, you don't make this hairpin. You make the one that the RNA polymerase doesn't care about. You don't make the one that is bad for the RNA polymerase. And because of that, the RNA polymerase stays on the DNA. It continues to transcribe, and it continues to make the messenger RNA that corresponds to all the enzymes that make that. Okay, so why do we call this attenuation? Because the amount of tryptophan is going to determine the amount of transcription of the genes. If the level of tryptophan is low, but not absent, then you're going to get a little bit of tRNA, and you're going to get a little bit of this leader peptide, and you'll get a little bit moving forward. But if the level is super low and you have these empty spots, then the ribosome will completely stall, and you'll have complete transcription of the gene. So what's going to happen is the bacterium is going to be somewhere between these two. If you have more tryptophan, you're going to be closer to the top. You'll have less gene transcription. If you have less tryptophan, you'll be more towards the bottom and less gene transcription. Attenuation. The fact that messenger RNAs can form secondary structural elements is also an important control mechanism for gene expression. And we'll demonstrate this with the heat shock proteins that was first discovered in E. coli, but it's what bacteria have. And heat shock proteins are going to be a set of proteins that are produced by bacteria when they're exposed to a high temperature environment. High temperature causes proteins to slightly denature. It can make membranes leaky. It can affect overall cellular processes. And these proteins are produced. They bind and stabilize proteins and structures to give the bacterium time to recover from the heat shock and to repair the damage. So clearly you don't want to make heat shock proteins under normal conditions. You only want to make heat shock proteins when you've had a heat shock. So we need to have a way to control the transcription of the gene. So here's the gene up here at the top. Here's the encoding the heat shock proteins that are over here. And of course, you'll have a promoter off to the left side over here. Here's the Scheindel-Garneau sequence that you see. Okay, so the heat shock protein is under control of the sigma 32 factor. Now, if you remember from the RNA synthesis or the gene transcription lecture a few lectures ago, the sigma factor will bind to the RNA polymerase, and together that sigma factor will help the RNA polymerase find a very specific sequence on the DNA so the RNA polymerase can transcribe very specific genes. And the sigma 32 factor is the one that controls the heat shock gene expression here. So when When the bacterium is exposed to high temperature, of course, sigma factor 32 is going to be present. It's going to help the RNA polymerase bind, and you'll see production of the messenger RNA. Now, an interesting thing about this messenger RNA is its structure is such that as it comes off of the RNA polymerase, each piece is going to have a complementary piece further down. So here's the 5 prime, you go all the way through, here's the 3 prime. And as that messenger RNA comes out, you'll see base pairing takes place with the first part, and then you'll see base pairing in this region over here, and then you see base pairing from the early parts to the late part. This is secondary structural elements. Now, the small ribosomal subunit is looking for the Schindel-Garno species. That's how the small ribosomal subunit knows where to bind. And it positions the ribosome so that that AUG site is right where the protein synthesis will take place. Now, because you have double-stranded RNA here, the ribosome can't bind. If you recall, ribosomes have RNA in it that is complementary to the Schindel-Garno species that allows the messenger RNA to bind to the small subunit, and that can't happen because of this double-stranded arrangement here, okay? So under these conditions, when the temperature is lower, the messenger RNA, if it should get transcribed, is going to be in a form that will not be translated by the ribosome. There's no translation. That's at fairly normal temperatures there. Now, when the temperature rises above 30 degrees, of course, temperature's low, you're not getting a lot of gene expression. But when the temperature goes up and the sigma 32 takes over and you start getting expression, gene expression here, then the messenger RNA, because the temperature is higher, you will get melting. And just means the temperature is high enough, there's enough thermal energy to break this these base pairing arrangements here, and you'll get a linear messenger RNA. And because it's linear, the small subunit can find the Schindel-Garno sequence, and then the large ribosomal subunit can bind, and you'll get protein expression. So as long as the temperature stays high, as long as this messenger RNA stays linear, the ribosome can continue to translate and make more heat shock proteins. But as the temperature drops below 40 degrees, you'll start to see these pieces come together and form into the secondary structure, and that will essentially turn off translation. And at the same time, of course, we'll see less sigma 32, so we'll see less mRNA production together. This concludes the first part of the lecture for prokaryotic gene expression regulation, and stay tuned for part 2. And welcome to part 2, which is the regulation of gene expression in eukaryotes. And from this little sneak peek here, you can see the level of complexity for gene regulation is much higher for eukaryotes, which of course matches their higher level of organization that they have. For the discussion of eukaryotic cells and gene expression regulation in eukaryotes, it's best just to focus on the human condition, because this is, of course, relevant to human medicine and the human condition. So what we know for human cells is we know that all of our cells, almost all of our cells, are going to contain an identical genome. We have the same chromosomes in all the cells. But we know that there are muscle cells and nerve cells and liver cells and kidney cells and bone cells. And because of that differentiation, there are going to be some genes that are expressed and some genes that are not expressed in any given cell. In fact, only a small fraction of the genome is going to be expressed in any given cell because of that. It has to do with what proteins do you need if you're a muscle, or what proteins do you need if you're a nerve, and that's going to control the gene regulation. But there's also constitutive proteins that are like, maybe you need more glycolysis in this cell, or maybe less glycolysis in that cell, or maybe more structural proteins in this cell or less. Again, the gene regulation is going to be matched to what the cellular function is. And that's going to drive the expression of gene transcription for eukaryotes. Here are the major gene expression regulation steps that you'll see in eukaryotes, and certainly in humans as well. Because there's a higher level of organization, there are subcellular organelles, and eukaryotes can do a variety of things that prokaryotes can't do. Now, one thing that is most striking is in the prokaryotic organism, in the human, is we know that our DNA is going to be organized into ultrastructure that forms chromosomes. And there's going to be chromatin, euchromatin, and heterochromatin, depending on how open or how closed that the structure of the chromatin is. And so it turns out for eukaryotic cells, the structure of the DNA and whether it's packed into the euchromatin or the heterochromatin and chromosome structure, the availability of the genes is the major mechanism that controls whether or not we have gene transcription. And that's different than the prokaryotes. Prokaryotes have simpler DNA. They don't have the ultrastructure that eukaryotes have, and so for them, it's whether or not you transcribe the DNA or not. That's their major mechanism. So, but for eukaryotes, it's the exposure, it's the availability of the DNA, it's the availability of the gene that really controls the transcription. But like the prokaryotes, we also have transcriptional control, whether or not the RNA polymerase will bind, whether or not it will transcribe, whether it's more active or less active. Some of the things we saw before, repressors and activators, are going to be the same for the eukaryotes as well. But we can go a little bit further. Because eukaryotes have nuclei and the DNA is contained within the nucleus, then once that mRNA is formed, it's formed within the nucleus away from where the ribosomes are. And that lets the mRNA live longer and it can be processed. We can see differential splicing and we can see organization. We can do things to the mRNA that the prokaryotes can't do. And then we can change how that mRNA exists out in the cytoplasm. We can change the stability of the mRNA and whether or not it's in a form that the ribosomes can bind to and translate. So there's going to be translational control as well. And then the last thing we have is we're going to have post-translational modification of proteins. And that would be proteins that are formed in a pre-form that are not active, and then they get processed to a mature form, which is active. Or it could be post-translational modification, where you add a fatty acid or a carbohydrate or something like that. So these are the major mechanisms that we see in the eukaryotic cells. All right, if you remember, there are two major forms of our chromatin being the DNA. And when the DNA is condensed, then it's called heterochromatin, and that's what you're seeing over here. All the DNA is wrapped around these nucleosome cores, and it's all packed together. And then when the chromatin is open, or when it's free, it's going to be called diffuse or euchromatin. That's typically when genes are accessible, and that's when you can make, do gene expression and make the mRNAs. And whether or not you're in heterochromatin or euchromatin, it's going to depend on the level of gene expression, and that's going to come down to what the histone core proteins are doing. So obviously there's going to be a process, gene regulation, driven by the interaction of DNA with the histones. Let's see how that works. You may recall from a couple of lectures back that when you have a histone core, histones are going to be proteins. It's going to be an octamer of these proteins, and they come together and they form these little kind of circular blocks, little cylinders that have a large amount of positively charged amino acids on the outside. And those positively charged amino acids will interact with a negatively charged phosphate backbone on the DNA. And when you look at the crystal structures, the proteins are shown in color, and then the double-stranded DNA is shown around here, and you can see it flat on, and you can see it from the side on, you can see it from the top as well. And we see that the DNA is wrapped very nicely, 1-2 turns around each one of those. So this, one of these things is called a histone core. And then there's a piece of linker DNA, and then the next histone core. So while this is more open, it's still not open enough. The RNA polymerase can't bind. It can't work on the DNA when it's interacting with the histones. So we have to go further than this if we want to have gene expression. And the way that works is that the histone proteins are going to have some amino acids that can be modified. There's lysine residues, they're positively charged. There's arginine residues, they're positively charged. And there's serine residues that are neutral. Serine is OH, it's neutral. And when the amino acids have their positive charge, that's going to allow them to interact with the negative phosphate backbone of the DNA. And when the serine is neutral, it will not interfere with the close association of the DNA with the histone core here. But we have specific enzymes that can acetylate lysine residues, or they can methylate the lysine residues. That will put chemical groups onto the end of the lysine side chain or the arginine side chain that will remove the positive charge. It blocks the positive charge, removes it or blocks it, so that you no longer have those electrostatic interaction. So when you change these side chains, then they no longer will interact with the DNA, and the DNA will actually unwrap, and the DNA histone proteins will fall apart. They will leave the DNA alone. The other thing that can happen is you can phosphorylate the serine. You put a PO4 on that oxygen, and that introduces a negative charge on the histone. And when that happens, you have a repulsive effect, where the negative phosphate repels the negative phosphate backbone. And so one of the things that happens to the cell is the cell says, I want to transcribe genes. It's going to activate the enzymes that will acetylate or methylate those proteins. And then when you want to repack, you deacetylate or demethylate. So the enzymes are shown here. I'm not really sure if the MCAT goes that far, but I do know the MCAT will ask about acetylation, methylation, and phosphorylation. And it's oftentimes in terms of the writers, these are the things that change the histone proteins. The erasers undo those changes, and it tells you what the effect is on the DNA. So here's the process, and what we find here's a histone protein that has a lysine side chain, and that lysine side chain has that terminal amine group, and it has a positive charge. And as a positive charge, it's able to interact by an ionic interaction with the phosphate backbone of DNA. So when the cell says it's time to unwrap that DNA, it's time to break those ionic interactions, it will activate a histone acetylase or a histone methylase enzyme that will modify. In this case, we're seeing acetylation. So here's acetyl-CoA, there's the acetyl group, and that acetyl group is going to be transferred onto the lysine, as you see there. So what happens in this case is you lose that positive charge. And because you lose a positive charge, you lose that ionic interaction. So the HAT enzyme is histone acetyl transferase. HAT is the enzyme that acetylates. I think that's MCAT knowledge. I think knowing what the HAT enzyme is does that. So as long as the lysine is acetylated, you have that neutral charge, and there's not going to be a positive ionic interaction with the DNA. When the cell says it's time to wrap the DNA back up, you want to move back to the packed form, then you're going to use a histone deacetylase enzyme. So H-histone deacetylase enzyme, HDAC, that removes the acetylate from that lysine amine group, and of course it gets protonated and becomes positively charged again. That allows the DNA to wrap around and form those ionic interactions like we saw before. So this is reversible. You acetylate and deacetylate, and that changes the exposure of the DNA. So the way it works is it's pretty complicated, is that you'll have the DNA all wrapped around these histone cores. These are nucleosome cores. You see the linker DNA in there. And there's going to be a factor, and the factor is going to be just a protein that is sitting here. And that factor then is going to activate the enzymes that will work on the side chains. So when this factor is present, when it binds, it's going to recruit these co-activators to come, which will then modify the amino acid side chains, which will then cause the histone cores to fall apart. The DNA will unwind. You'll have this long linear piece of DNA. And what we see here is the RNA polymerase is sitting here waiting to go. It's ready to go and start transcribing the genes as soon as we get this. And of course, the co-activators will also talk to the RNA polymerase and get it started as well. So the little bit more, I think, I don't know if this is really MCAT knowledge, but the way it works is the RNA polymerase is here and it's ready to start transcribing these genes, but it can't until something tells it to do so. And so we see this folded nature of the DNA, which brings these modifier sequence together and these activator proteins together, which is going to bring about the change in the structure, the ultrastructure of the DNA, which will unwrap unwrap and allow that DNA to become linear so the RNA polymerase can do the work it needs to do. Another change to the DNA that affects its ability to interact with the RNA polymerase is methylation of DNA. And here we're showing specifically the methylation of cytosines that are in the DNA structure here. And there'll be a slide, I think, showing this, and we've sort of covered this a little bit earlier with the structure of the cytosine, for example. But when you methylate this, you really change the hydrophobic nature of that cytosine and how well this thing interacts with the environment around it. So when you get multiple methylations in an area of the DNA, that's going to change the local organization of the DNA or the packing. It causes a reversible and temporary change in the gene status by causing a change in the overall structure of this DNA. We'll see it on the next slide. Now, sometimes the methylation will be permanent. For example, during fetal development, when organs are forming and the body is forming, there's a lot of genes that are turned on that are responsible for developing the fetus into a fully functional, ready to be born baby. And then after birth, you don't need those genes to be active, and they'll be methylated in a permanent way that keeps them turned off forever. But there's also reversible DNA methylation, which can downregulate a gene if necessary. And we're not going to split those hairs for now, but it's a couple of ways that's done. And the way it works is that when you have methylation of that cytidine, it's going to change the hydrophobic nature, which can change the B form of DNA into the Z form of DNA. Now, B form is a little bit more exposed, more active. You can see how the more open structure here, it's easier for this to be worked on by the RNA polymerase, for example. The Z form, because it has a tighter twist, it's going to be more tightly packed. The RNA polymerase can't bind and it can't unwind those two strands, so it can't do gene expression. And methylation is one way that drives the B to Z transition. So another way that genes are available for transcription depends on chromatin remodeling. And what that means is there may be a gene located on a chromosome that may be in a less accessible area. And then by gene rearrangement, that gene gets moved out to an area of the chromatin that is more accessible. And gene rearrangement will then move elements in and out of exposed areas that change the overall gene transcription. A really great example is the way antibodies are produced by B cells. Now, this is very much an immunology topic. For the OMAPS class, immunology doesn't come until later, so try not to focus on the immunology. Try to focus on the protein structure function based on what these genes are doing. Now, what we know is that there are millions and millions of different antigens and millions of different antibodies. And because of that, there has to be all kinds of B cells that make these different antibodies. And just the structural complexity and diversity just sounds like an overwhelming problem. Well, nature figured out how to fix this because when you have an antibody protein, and you remember this from the earlier parts of this biochemistry class, for the antibody part, there's going to be constant or conserved regions of this antibody, and that's going to be part of the antibody structure that doesn't change. But then you're going to have variable regions in the antibody molecule that does change. And so maybe we can set this up so that these proteins, these constant sequence proteins, are produced constitutively with the gene that's more exposed, and then all the cell has to do is just make this variable region. And so by doing selective gene rearrangement, you can make the pieces that are needed in order to make the antibody for this particular B cell that goes after a particular antigen. So let's see how that works. So it's really complicated, and I don't want to do a deep dive on this, but what we find is in an undifferentiated B cell, the B cell says, well, I need to have certain parts of the antibody molecule because I know no matter what the antigen is, I'm going to need those fundamental parts of that antibody molecule. And so when the B cell differentiates, it's going to cut out stuff that it doesn't need, but it's going to keep the stuff that it does need. And you see here that parts of that DNA are going to go forward. And as you get to the final stage, when the B cell is fully tuned to whatever it is that the antibody that it needs to make, you will have this final piece of the DNA here. And that DNA now is going to be transcribed into an mRNA that gets processed. We'll talk about that a little bit later, until eventually you have the final protein. And so it turns out that you really only end up making a very small bit of the protein. Only a small bit of the DNA is really needed in order to make that specific antibody. And so by rearranging the genome, by rearranging these elements, you can come up with a large variety of proteins from a small number of standard pieces. Let's move now to gene expression regulation at the level of transcription. And as I mentioned a couple slides ago, this will be very similar to what we saw in the prokaryotes, where you have operators and modification of the promoter site and so forth here. And when we look at this, we're looking at the DNA, and then we see the initiation site. If you remember, for the eukaryotes, the TATA box, there's a couple of them, but TATA is one of the better understood. And there's the TATA binding protein that recognizes that specific bit of the DNA. And of course, the TATA binding protein is what allows the mRNA, the RNA polymerase to know where it should bind so it can start the transcription process. So in that regard, that's where we've been before. But now where you see the complexity coming in is there's going to be a variety of transcription factors here that will enable the RNA polymerase to do its work, but also regulate the RNA polymerase. We see here a trans-activating factor over here at this site. And up here we see a hormone receptor. So we're going to talk about the hormone receptors first here. Hormone receptor, and what these different molecules are is each of these pieces that bind to the DNA are going to control the ability of these proteins to come together. And when they come together and they interact, they make what is known as a functional activation complex. Any one of these things by itself can't make it happen, but when they're all together doing their work, then the RNA polymerase gets the message that it needs, that it's okay to go ahead. So let's start first with the hormone receptor, because a lot of eukaryotic gene expression at the transcription level is going to be driven by hormonal signaling. This is a slide we saw back in the cell signaling lecture, actually part 2, the second hour, where we were talking about intracellular receptors. The intracellular receptors exist out in the cytoplasm, or they'll be inside the nucleus. And the idea is the hormone will come to the cell membrane, find its way across the membrane, and bind to a specific receptor. In this case, the cytoplasmic receptor is going to be held inactive by what's known as a heat shock protein. Talked about those, but and when the hormone binds, it causes a conformational change in the receptor that makes it let go of the heat shock protein, and this activated complex now is then trafficked into the nucleus, and that receptor has a very specific structure for a very specific bit of DNA, and that's usually called the response element. So whatever hormone the receptor binds and into the DNA is going to be what the name of that response element is. So for example, if we're looking at a glucocorticoid hormone that binds to the glucocorticoid receptor, it will bind to the glucocorticoid receptor element. And one of the points we made before is that these receptors almost always will form dimers. Heterodimers are different. Homodimers are the same. Kind of depends on what intracellular receptor you're talking about. But it takes both receptors to be present in order for them to have the weak interactions between them and sometimes catalytic activity. But then these hormones will allow the RNA polymerase to become active and do the gene transcription. The other type of intracellular receptor is when the hormone comes into the cell. If it's going to bind to a nuclear receptor, it will travel all the way into the nucleus, where it will find its empty receptor. When they bind together, again, there's going to be a conformational change, and that will allow this receptor now to have a conformation that has high affinity for the DNA. It binds to its DNA response element. Again, we're looking at a dimer. It could be a homodimer. In this case, you're seeing a heterodimer, in this case, 2 different ones. And it really depends on what the hormone is and what the DNA is. But again, when they're bound to this response element, that is going to allow the RNA polymerase to get the information it needs that it's okay to go ahead and transcribe the gene. So hormone-dependent receptor signaling and then DNA gene transcription. Now up here a little bit different. I don't know if we showed this in the signaling lecture, but what you see is for these intracellular receptors, they're going to have part of the protein. This is the protein structure, N-terminus on the left, C-terminus on the right. There's going to be a part of the protein that binds the ligand. There's going to be a part that makes the general structure of the receptor, and there's going to be a specific DNA binding domain here. So depending on what this variable region is determines whether it's a homodimer or heterodimer, and of course the DNA region determines where it binds. Okay, so that is the intracellular receptor, or the hormone binding receptor part. Let's go down here at transcription factors. Now again, remember a factor is going to be a protein that binds. And so you'll either have activators or repressors. And so in this case, it's showing you that you have co-activators. These are going to be proteins that help recognize the TATA binding protein. The TF is going to be a transcription factor. They're numbered based on what their function is. So transcription factor A, B, C, they all have different functions. You don't have to memorize that, but just recognize that there are a variety of proteins that are going to work together. All of these have to be assembled. All of them have to be functional. And when they are going to equip the RNA polymerase to do the kind of work it needs to do. Now, this just shows these proteins parked at the DNA, but there's actually a fairly sophisticated structure that allows these transcription factors to bind to the DNA. And these are the motifs, and I'm not absolutely sure, but I think the MCAT might ask you to at least understand what these structural motifs are. So what you'll find is there's a zinc finger, leucine zipper, helix turn helix, and helix loop helix are the motifs that we have. And what that means is that these are proteins, their size and their shape and the charged amino acids are going to be perfect for them to fit either in the minor or the major groove, usually the major groove. And by getting into that major groove, it's going to let them snuggle up to the DNA and bind to a very specific DNA sequence that will be at or near the TATA binding protein that will help localize that RNA polymerase. You see a very sophisticated protein DNA interaction going on here. So whether it's a zinc finger, the zinc ion just is a positive center that allows this thing to fold with a certain geometry that lets it bind to the DNA. A leucine zipper just means there's two helices and there's leucine residues or isoleucine residues, and they form hydrophobic interactions. And you see they're both going into the major grooves for the two halves of the DNA double helix there. And again, these are going to be proteins that are going to allow other proteins to bind. Helix turn helix, where you have one in the major groove and then two that lay down on the outside. And of course, a combination of all these together, you have these two, typically a leucine zipper here with more protein. You don't have to memorize this structure, but just recognize the names because I think that's going to be important for MCAT. Now, this is something we've seen before. This is very much like the prokaryotic system where we're looking at repressors and we're looking at activators. We're looking at negative control and positive control. And we're going to see trans-acting factors and we're going to see cis-acting elements. So what we'll see is there's going to be a regulatory protein. This is going to be a protein that exists in the cell. It was coded by a different piece of DNA. It had its own messenger RNA. It's a protein. And when it's in the nucleus, it will have a very specific site where it will bind. And by binding here, the picture is showing you that gene A is producing messenger RNA. So right away, what you say is this must be a positive control. But obviously the thing is binding, making it possible to do gene expression. Now, the cool thing about eukaryotes is that a lot of times this regulatory protein will bind to a number of sequences here for a number of genes that are related in function. So in this case, when you want gene A activated, you want gene B activated. So you'll see this having a dual function. So these mRNAs are going to do things. Now in this case, the single mRNA for gene A is going to have some function, don't know what it is, but you need to make that function. Now over here, gene B is going to make a protein that itself is going to become a modulator of these other genes as well. So you see maybe these are going to be different enzymes as part of a metabolic pathway or proteins that make up a structural unit, something like that. And so again, the cell, because of the complexity, you can get gene A activated, you can get gene A and B. There are times when you could see mix and match of whether there's an inducer or whatever. It's really complex how it works. But generally speaking, all of these genes are going to have a related function, but whereas the bacteria have these genes 1, 2, 3, 4 in a line with a polycistronic mRNA, these genes can be separated over different chromosomes in completely different areas, and you're going to see activation of each of these genes in the background of whatever the genes are that are around, and you're going to get that concerted action. Really, really nice. All right, the third mechanism we made the point that gene transcription takes place inside the nucleus, and the nucleus, of course, has its membrane, so it's a separated organelle from the cell. So when the mRNA is made, it can be held in the nucleus for a period of time, which gives the cell time to work on that messenger RNA, and it can change the structure of the mRNA. And so we'll develop that a little bit now here. So what we see is we see that there is the DNA, and from the DNA, the messenger RNA is going to be made. And if you recall, there are exons and there are introns. And exons are protein coding sequences, and introns are non-protein sequencing sections. And when we make the mature mRNA, then you cut out the introns and you're left with only the exonic that leads to the protein formation. Now, what alternative splicing means you can mix and match depending on which pieces of the protein you want. So for example, here on the left, there is 1, 2, 3, 4, 5. All of the introns are taken out, and so you're left with 1, 2, 3, 4, 5 exons, and that's going to give you a particular protein with a particular structure. Now, it may be that you want to have a similar function, but you want to have slightly different activity, and so you want to remove part of the protein, but keep the other part. And here you see 1, 2, 4, and 5. Section 3 does not appear in this protein, and that's going to give an overall different fold and a different function. Or over here, you might lose that last loop, but keep the intermediate one. So you have 1, 2, 3, but not 4, but 5. And so by alternatively splicing, you can make isoforms or different members of a protein family that will have different function. You may have just a ligand binding that's not present, or you may have a voltage that may not be. All kinds of stuff can happen with this alternative splicing. Very complicated. Again, we could spend several lectures just talking about alternative splicing. So it's a little bit hard to see this, but there are 5 kinds of alternative splicing, and you'll see that there are parts of the protein here. This blue cylinder on the left side, the blue cylinder on the right side are going to be preserved. So that's what's called a constitutive exon. Whatever this protein is, you always want those ends to be the same. But then you can have a lot of differences that take place depending on what part you keep and what part you lose. So you see there are intronic sequences. There is also an exon here that may be kept intact or may have different pieces with it as well. So I won't ask about the specifics here. I don't think the MCAT will ask about this, but it's important to understand that alternative splicing is a way to keep certain parts of the gene encoded mRNA, and you splice out or remove certain parts, and that's going to change the final protein product. This is the same information that was on the last slide, but this shows you 5 different types that are there. Next form of processing we have is called mRNA editing, and it's exactly what it sounds like. You'll have a gene, a DNA, and that DNA will lead to the production of an initial mRNA transcript. And so it'll be a preform or a not yet mature form, and there'll be a special area inside this that can be modified. So if you're a cell type and you need the full-size protein that goes with a full-size mRNA, there will be no editing. The mRNA will be finalized, and you'll have the mature mRNA, and that'll lead to a full-size protein. Okay. But there may be another cell type that wants to have a similar protein, but not as big of a protein. And they will edit a codon that will create a stop codon. In this case, the C becomes a U, UAA is a stop codon, and the mature messenger RNA will be translated by the ribosome, and when it comes to the stop codon, the ribosome will stop making the protein, and you get a smaller protein product. And so the example here are liver cells and intestines that make the protein ApoB. And in the liver, the protein is called ApoB100. It is larger, it is the full-size protein. In intestinal cells, they make ApoB48. ApoB48 is a smaller protein, but it has a very similar function to ApoB100, but there are important differences by how these proteins functions, and we'll cover that in a couple of lectures coming up. Let's talk about regulation of gene transcription at the level of translation. And here we're going to change the initiation of translation. Literally, that means the mRNA has been produced. It has now been released to the cytoplasm. And the question is, can the ribosome assemble onto that messenger RNA, and can the ribosome then start to translate and to make the protein? And so we're going to see a couple of mechanisms that affect the ribosome's ability to interact with or to translate the mRNA. And here's a little bit of a spoiler. If you look, the eukaryotic initiation factor 2, EIF2, and it's a GTP-dependent protein, and it has a pretty big role. So let's look at this. Let's see how this works. Let's see how we can regulate gene expression at the translation, the initiation of translation in this process. And to do this, we're going to look at globin synthesis for making the molecule hemoglobin, which is going to be in red blood cells. And of course, before a red blood cell is formed, you have a reticulocyte cell, which is an immature red blood cell. And one of the jobs of the reticulocyte is to make a lot of hemoglobin so that the red blood cell will be full of the protein when it gets released. So to do that, you need to drive quite a bit of globin synthesis to have the subunits needed to assemble the hemoglobin. All right. Well, if you remember hemoglobin, there's a tetramer, there's 2 alphas and 2 betas, okay. But we know that each one of these subunits is going to have a heme group inside of it. And so you want to make sure that there is enough heme to match with the protein that's being synthesized. And you don't want to synthesize the protein if you don't have the heme. So obviously heme is going to be an important signal or important regulator for globin synthesis. And in fact, that's the case. Globin synthesis is controlled by the amount of heme that's available. When heme is available, globin synthesis is going to be active. When heme is not available or low, globin synthesis is going to be not active or less active. Well, how does that work? Okay, well, let's pick up where we left off before. The eukaryotic initiation factor 2 alpha. That's going to be one of those factors that interacts at the RNA polymerase binding site for the globin gene, alphas and betas and so forth. And that's one of those factors that when it's active allows the mRNA, the RNA polymerase to bind and make the mRNA for the globin protein. Now this factor has an active form and an inactive form. And the inactive form is going to be phosphorylated. The active form is not phosphorylated. So who does the phosphorylation? There's this protein called the heme regulated inhibitor kinase, or HRI kinase. And it is going to have as an allosteric modulator the heme molecule. And that's what's shown up here. Heme is an allosteric modulator for this kinase, and if the kinase is active, it will phosphorylate EI2, and if it's not active, it will not phosphorylate. If you remember from the signaling lecture, when a kinase is active, you have a phosphorylation status. When a kinase is not active, there is a phosphorylase that will pull the phosphate off. So when heme is present, heme is going to inactivate the HRI kinase because it wants to make sure you don't phosphorylate this initiation factor. Remember, when it's phosphorylated, then the initiation factor doesn't work. You can't get globin synthesis. So heme will shut down that kinase. And if the kinase is shut down, the phosphatase is turned on, and you'll get dephosphorylation. When heme is low, when there's not heme in the cell, then you're not going to have heme bound to this HRI kinase, and so you won't have the inactive form. Instead, you'll have the active form, and that kinase will then grab and phosphorylate any EIF2 and make it the phosphorylated form. So when there's no heme, you will not make globin. When there is heme, you will make globin. And so that's what we mean about the initiation of translation. If you look in the slide before this, when you saw that EIF2 alpha, it's the one that's going to get with that small ribosomal subunit and allow the process to get started. So here's a different type of regulation of initiation of translation. And in this case, there is a messenger RNA, and that messenger RNA has structure that allows it to form base pairing interactions that make a little hairpin. And then that hairpin itself is going to be recognized by a protein, a very specific protein. So that mRNA is called the response element, and the protein is going to be the response element binding protein. And when that protein is bound, it physically blocks the AUG initiation site. So the ribosome can't bind, and the ribosome can't translate the mRNA and make a protein. But there will come a time when a ligand will bind to this response element protein and cause a conformational change. And when you have a conformational change, the protein comes off of that hairpin. And when there's no longer that bulky protein sitting there, the ribosome can find the AUG, and then it can start doing the translation and making the protein. And a really good example of this regulation strategy is for the synthesis of the protein ferritin. Ferritin is a protein found in cells, and its job is to store iron. It binds iron to keep the free iron level low inside the cell. We'll see later in the biochemistry class that free iron can cause the production of free radicals, and the cell doesn't want that. So the best way to do that is to keep iron tightly bound inside the ferritin where it's protected and it can't form free radicals. So when iron levels increase inside the cell is going to say, I need to make more ferritin to make sure I can bind that iron and keep it complexed. And in fact, that's what happens. Normally, ferritin synthesis is going to be downregulated by the iron. This is an iron response element, and the iron response element binding protein, it's going to block the synthesis of the ferritin protein. When iron levels start to rise in the cell, iron will bind to that specific binding site, causing a conformational change. The iron response element binding protein comes off, and then the ribosomes can now bind, and it can synthesize more copies of the ferritin protein. And as more copies of ferritin protein become available, it binds the iron. The free iron level inside the cell drops. Iron comes off of the iron response element binding protein, and it allows it to bind back to this hairpin loop, and that stops the synthesis of ferritin. So it's a really nice, very sensitive way of handling this situation. So the next one is going to be the stability of the mRNA. And this is really complicated. There's a lot of steps here. This is a giant field for how DNA and RNA work, but also for therapeutics. Silencing RNAs, microRNAs is what we're looking at here. And so all I can do is really just point to what this is and give you some of the big picture here. But an mRNA is going to be present in the cytoplasm, and it's going to have a lifetime associated with it. And that lifetime, if you remember, eukaryotes put a 5' cap, and they put a poly A tail on the 3' end, and that greatly extends the life of the mRNA. And that gives the eukaryotic cell more time to process that mRNA. But there's other factors that can affect the stability and the lifetime of the mRNA as well. And these are going to be called small RNA molecules or microRNAs. And those microRNAs can interact with the messenger RNA in a way that can either change the accessibility of the ribosome for translation, or it could put the mRNA in a form that makes it less stable. There's a lot of things that can happen. And that's why we can't really talk about it too much here, because there's just too many options. But what we find is that the the gene now is going to make the primary mRNA construct, and that will go out like we've been talking about so far. For genes that are going to be modulated by these microRNAs, the microRNA sequence is usually proximal to the actual protein coding gene, so that as the RNA synthase is making the mRNA, it will also make these small microRNAs. And you see it synthesized in a preform first, and that preform is going to be processed in the nucleus to its final form, the pre-microRNA that gets out into the cytoplasm. Now, whether or not this is active is going to depend on some processing that happens afterwards. And there's a protein called dicer. And dicer will bind that pre-microRNA and process it so that it has a final structure. And that final structure usually is going to be a dimer. It's going to be a base paired dimer here. And so that microRNA itself is not going to be active because that base pairing prevents it from interacting with the non-base paired mRNA. So there are other proteins here, in this case the RISC protein, which is an RNA-induced silencing complex, RASC. And the RASC will bind this base paired microRNA, pull it apart, and then expose that non-base paired part, and it will bind to a specific sequence on the mRNA. And by doing that, it's going to form that double-stranded mRNA or the double-stranded RNA molecule here. Again, that's going to inhibit the progress of the ribosome. And if the ribosome can't move to make the protein, of course, that's going to mean the mRNA is going to be eventually degraded and you won't get the protein translations, what this is saying. So either you stop the repression for a period of time, maybe the risk comes off, or maybe the thing gets processed. And then the fifth one is going to be post-translational regulation, which is really cool. And students find this a bit confusing, and I did for a while as well. When we think about gene regulation, the gene expression regulation, we're thinking about DNA, we're thinking about RNA, we're thinking about messenger RNA, we're thinking about all these things we've been talking about. It's really hard to imagine how talking about a protein is gene regulation. But remember, the idea of gene regulation ultimately is I want to change the proteins that appear in the cell. And whether they're proteins, whether they're synthesized or not, or whether they're isoforms or family members, whatever, all of these things matter about the final protein. And so if you have a protein that can be synthesized in a pre-form, a premature form that is not active, then you have the ability to modify that protein to its final mature form. And by making it in its final mature form, it will have the function or the structure or the enzymatic activity. You will change the ability of that protein to do what it needs to be done. And so that is post-translational modification of proteins ends up as the final way you can regulate a gene product to change the cell. And the best example of that is going to be enzymes that are synthesized in a preform that's inactive called zymogens or proenzymes. And you see they have a little bit of an extra amino acids that's going to cause the protein fold to go into a form that's not really very active. We talked about this during the enzyme lectures. And when this enzyme is released and it's put into its site where it's going to be active, another enzyme, another protease enzyme will work on this, and it will selectively remove these couple of amino acids. And when those amino acids disappear, it's going to cause a reorientation or a refolding of the protein to accommodate the loss of those weak interactions. It's hard to see, but you can see this green loop is gone because it's been cut, and you can see this loop used to go up to sort of up to the top, but now it goes up to the left. And if you look, you see other areas that are different. You see loops are different, and it's the protein is folded differently. And now the enzyme active site is exposed, and this protein is active. And we've also seen this with like signaling proteins, for example, insulin. Insulin is synthesized in a preform first that is non-active and cannot bind to the receptor. But for it to be active out in the blood, it has to be in its final mature form. So when insulin is synthesized as a proinsulin in a pre-form, and it gets stored in those pancreatic beta cells, then you'll cut off this part of the protein called the C-peptide, and you're left with the mature form of the protein that will adopt its final fold, and that is the active form of the protein, and it will be released in the blood when it's released from the beta cells in the pancreas. And this is a strategy that's used for a variety of hormones that circulate through the blood, these endocrine hormones, if you would. Okay, a couple questions if you'd like. Two questions on the same one. It must be 2 for 1 today. So which of the following best describes regulatory proteins that prevent the binding of prokaryotic RNA polymerase to the promoter, in this case the promoter in the operator region, and thus act on the initiation of transcription? Basically, what is it that blocks transcription? Do you see one that works here? Okay, maybe. So an inducer usually induces, it causes something to happen, and I recall the repressors had an inducer, so you might want to hold on to that one for a second. DNA binding proteins, no, I don't think so. DNA binding proteins are going to be found in the eukaryotic system, and they're going to work together with that large complex. A repressor, yeah, repressor sounds pretty good, because if a repressor binds, it's going to block the binding of the RNA polymerase and not let it do it. So let's put a note on that one too. So now we've got A and C. Sigma factor? I don't think so. I think sigma factors are what bind to the RNA polymerase that let it find the exact sequence of DNA where it will bind and start transcribing the DNA. So the sigma factor is not going to repress or not going to prevent the transcription. Transactivator? Wow, so a transactivator, that's usually that the transacting factor that comes in, it binds the operon, maybe. Okay, so what the stem says up here is it's going to bind to the prokaryotic RNA polymerase and it's going to stop the initiation of transcription. So a repressor becomes the best answer now. The transactivator is not going to work because even though it's a trans factor, it has to be a trans repressor. It doesn't activate the genes, it represses it. And why not the inducer? If you recall, the transacting factor will be in the cell but not bound to the DNA. When the inducer binds, it allows that repressor to bind to the DNA and stop the RNA polymerase. So in this case, it is the repressor itself that's going to bind as it's formed, and only when it binds a co-molecule will it actually come off. So we're going to see that one. And I guess I have to do both of them here. The ability of heme to affect synthesis of globin by controlling the activity of the heme kinase. Can phosphorylate eukaryotic initiation factor ELF2 is an example of what kind of gene regulation? Let's see, the eukaryotic initiation factor. I think that was going to allow the, oh gosh, what was that? The ELF, oh, that's right, the ribosome getting onto the mRNA, which allows translation. So that must be a translation-mediated regulation. And in fact, that's right. Repressor and translation.